All right, is fever. Now, fever is a little bit like inflammation. We tend to think of fever in a negative way because nobody really likes or enjoys a fever. But again, it serves a purpose. This is another example of a non-specific response to a challenge to the body. This is often going to be caused by, a bacteria or a virus that is circulating in the body, but could be caused from certain types of cancer cells and those sorts of things. But fever needs to have a trigger, just like inflammation had a chemical alarm. Fever has to be triggered as well. Now, does anybody remember what part of the nervous system was responsible for regulating body temperature?
Hypothalamus?
Yes, very good. Normal regulation of temperature occurs in the hypothalamus. We have cells in the hypothalamus that will respond to signals of either increasing or decreasing body temperature, and we'll get a response. So if the signal is that we're cold, right, our body temperature is dropping, the hypothalamus will signal skeletal muscles to shiver, and will signal hormones to increase, certain hormones to increase basal metabolic rate. And when we increase basal metabolic rate, we need more ATP. And when we use ATP for energy, we create heat, right? So that's the basis behind temperature regulation in the hypothalamus. But if the hypothalamus is signaled to change the thermostat, so what do we say a normal temperature is in the body, humans?
Is it 36.5 Celsius? Around there?
Yeah, it's around there. Okay, I'm not going to get too excited about it because we understand that there's a bit of fluctuation. It's probably a little closer to 37, even maybe 37.5. But at a given time in the day, it could be 36.5, it could be 37.5. And in reality, it might go up as high as 39 and as low as 36. And we recognize that somewhere in the middle of that is our number, is our average number or our plus or minus number, right? But we're not going to get too excited about taking a measurement at one point in time and seeing it at 39 or 38.5 and at another point in time seeing it 36. For example, if you've just run a race, if you just ran for a kilometer or 2 kilometers, you've been using a lot of ATP. Your body temperature is going to be high, but it would be expected to be high. If you are taking your body temperature first thing in the morning before you've started your day, your body temperature is probably going to be a little bit low. And then later in the day, a little bit high, just because you've been metabolically active, right? So we recognize that there's going to be some fluctuation. But in a fever, by definition, we're going to be up above our normal expected ranges. So that's going to be higher than normal temperature. And again, it's really not going to be defined as a fever if you spike at 39 or 39 and a half. But once it's sort of staying up in that range, it represents more of a concern. So fever, again, is initiated, the hypothalamus by pyrogens. Now, some of you are going to be aware of that terminology. Pyro has something to do with fire, right? Sometimes you hear, like in the movies and stuff like that, pyromaniac is somebody who loves fire. But a pyrogen is something that generates fire. That's a chemical that's going to be released by activated white blood cells. Some of the chemicals that get released in activated white blood cells, for example, neutrophil or eosinophil, are going to signal the hypothalamus to raise temperature. So the initial question might be, why? Well, think about this. So why viruses and bacteria access the body at a plus or minus 37 degrees Celsius. So if we raise, or even just say we change 37 degrees Celsius, let's keep our homeostasis in there all the time, becomes less favorable. Does that make sense? So the virus or the bacteria, it was attracted to you because your temperature was sitting, give or take, around 37 degrees. And as soon as you change it, in this case, bring it up, let's say you're bringing it up to 40, it's going to be less favorable environment for the survival of those viruses and those bacteria. They loved 37 degrees. All kinds of nutrition and environment that's favorable for them to be able to reproduce and access other cells if they need to, like viruses do. But now at 40, not so much. Right? There's some other mechanisms that are going on with this raised temperature. The liver actually starts to keep certain nutrients that are favorable for bacteria to survive with, but the liver starts to keep them, not make them available to them in the circulation. So they're not going to have a nutritional source. But the big thing, think about it in terms of the environment that they survive in. We're changing that environment. So what happens with these pyrogens is they encourage the temperature to change. So instead of being a homeostasis above and around 37 degrees, it might reset to 39. So now in a fever, our homeostasis looks like this, where we're at a 39 instead of a 37, say 2 full degrees warmer. And that means that our peak, we might be up here at 42 and down here at 37. So now during our homeostasis of the temperature, we're at a much higher average temperature. And that's what fever really represents. So how do you think you feel if normally we say you're at 37 and you operate, your muscles contract and your cells produce what they're supposed to produce and grow the way they're supposed to grow. But now the new temperature average is 39 and it might go up as high as 42. How do you feel? Are you going to feel cold or are you going to feel hot?
Very hot.
Oh, wait a second.
No, cold. My apologies.
Right? Because the thermostat is calling for 39, but your entire system is based on 37. So the adjustment, the signal is you're cold and you need to make adjustments to get hot.
Is that why we get the chills when we get?
Well, first of all, that's what shivers are. So the chills generate a shiver, right? And the chill, you feel cold because your thermostat has reset to a higher number, but your body is operating at the lower number. It's the same concept as when you go outside on a cold day and your exposure to the cold temperature is a signal that you better warm up because on that cold day, you're going to drop down below 36 if you don't get moving. And when you get moving, you're going to utilize ATP and generate heat. So the same concept is applying here. You don't have to be active for this to happen, but you are going to shiver in response until you meet the new thermostat, 39 plus or minus. Once you meet the new thermostat, you stop shivering. But most of your metabolic functions are based at 37. So now you're going to start to sweat. And when you sweat, you cool. But then your thermostat says you got to get back up to 39. So then you shiver. And you go through shivers and sweats, chills and sweats, which is what most of us go through when we're fighting an infection. If it's an infection that generates a fever. Right? So that's the mechanism. That's the reasoning behind this. Everybody okay with that? You sort of get that idea there?
Yes.
Yep. Okay. So let's finish up our innate immune responses by looking at a couple of chemicals or proteins that play a role in this non-specific response. So, for example, there's a group of proteins that are called complement. Okay. So these are a group of proteins that circulate in the blood, minding their own business, unless they get chemically signaled. They may get signaled from the activation of neutrophils or macrophages or these dendritic cells that we're going to talk about in a couple of minutes. But they are a group of somewhere between 20 and 30 proteins that are normally inactive, but when signaled, become active. So, like I mentioned, the signals can come from chemicals released from neutrophils or macrophages, or the inflammatory response generally will activate complement proteins. And when complement proteins activate, they kind of go in a sequence. So they're going to go from, protein 1 to protein 2 to protein 3, and will activate for as long as they're being signaled. So again, it's sort of a temporary positive feedback reaction. There's going to be this sequence of activations. So they are going to be activated by complements. The complement signals can include activation from these phagocytes, the eaters, from antibodies, which we haven't talked about because they're really part of the specific immune response, but they can activate complement. And what they literally do what it sounds like they do. They promote inflammation. So this is, again, part of the positive feedback. Inflammatory signals promoted complement action, and then the complement promotes inflammation. So it's a positive feedback and promotes phagocytosis. So again, complement is complementing the same things that initiated their response. Chemicals from the phagocytes stimulated the complement then stimulates phagocytosis. So positive feedback. We're going in a cycle in this case. And the other thing that they do, so these are so they can do that, they can do that. And then what they do, some of them will do, is they form what are called membrane attack. And the names sometimes in the immune system are great names. They're very violent. They're very much sort of, us against them type terminology. So we're going to form what's called a membrane attack complex. And they're abbreviated as MAX. But these membrane attack complexes are formed when complement protein binds to the antigen cell and creates a target. So that's sometimes known as being tagged. So the proteins bind to the antigen and attract other immune cells to create lysis. Lysis is a general term. A lot of times it's on the end of a root word, like cytolysis would be cell breakdown. Lipolysis, breakdown of lipids, right? In this case, we're talking about immune cells that are going to break down the antigen. So these are going to be enzymes that are released either by this group of proteins or by the attraction of other cells to the site of the injury or attraction of antibodies. And the idea is we're making a bigger target. By forming these membrane attack complexes, we make a bigger target for phagocytes and enzymes to break down the antigen. Okay, then we have another group of proteins that are called interferons. I mentioned them earlier, and guess what an interferon does? Take a wild guess at what function the interferon would do.
It doesn't interfere, does it?
It does interfere. So it's going to literally interfere with virus infection. So typically what's going to, or at least one mechanism for these, is that a virus infected cell by definition now becomes a non-self cell. So literally what happens, and this is what happens in coronavirus. So remember, I think we mentioned, maybe we didn't, in the coronavirus, it targets cells in the lungs. So in the coronavirus, we've got a receptor on one cell type in the lungs. So we're going to make this virus, we're going to make him an ugly looking structure. But what it's got is it's got this spike protein on it. And the spike protein will match with this receptor. So when the spike protein binds to that receptor, it causes the cell to change its behavior. And the cell will actually allow endocytosis of the virus inside the cell. The cell now is going to be exposed to the virus and it changes its behavior and it starts to make more virus. And eventually the virus fills up the cell and breaks the cell membrane, and the virus is allowed to escape and attack other cells in the area and repeat the whole process. So there's another group of cells with these same receptors on them. So now these new viruses infect those cells and kill those cells, but replicate and then make more virus. And during this time, you're going to have inflammation. You're going to have loss of function of those cells. Those cells have a purpose and their function has been lost, right? And the inflammation occurs because all of this new stuff taking up space is an irritant. And we're damaging cell membranes when they release, and we're creating this inflammation. So interferons, virus-infected cells can release a warning protein. And that's what's known as an interferon. Now, there's categories of interferons, but this is one of them. This is one of the early ones that was identified. So the idea is that if we're going to release, let's make our interferon, this virus-infected cell releases a chemical, protein, interferon, which signals these cells over here to be aware that there's a virus circulating. And in some cases, what the cells can do temporarily is they can make that receptor disappear. Or what they do is they can block that receptor. Let's make a so they can put a block over the receptor so the virus doesn't have a place to attach to. So these receptors are proteins, right? And they would be matched in this case to the virus. So a sneaky virus matches to a normal receptor in this case and causes infection. The coronavirus works exactly this way. Most of us have heard about this spike protein that's found on the coronavirus. Like they typically make the coronavirus look like it's this round thing with a bunch of spikes on it, right? And it would be three-dimensional, but those spikes are receptor finders, right? They're going to match up with a particular receptor, and the one cell in the lungs has a receptor for that spike protein. And that's why most of the symptoms that are serious symptoms affect the lungs. We're going to create inflammation, we're going to create damage to the cells, we lose some function of those cells, and then we also get the inflammation response, and we start to get our symptoms, coughing, loss of air exchange if it gets significant enough. That's where you end up in intensive care on respirators and that sort of thing. You following me okay with this? Does this make some sense?
Yes.
So the interferons are released from the virus-infected cell. It's sort of like, save yourself. It's too late for me, but I can give you a warning, and hopefully you'll be able to protect you. And the goal then is hopefully to prevent replication. Let's prevent infection and replication in nearby cells. Okay, so literally what happens is in a virus-infected cell, so if we said this was our normal cell, remember we said we had protein sequences on this cell that identified it as self. So let's say our sequence looks something like that. But we also have this receptor. And this receptor might match up with a spike protein. And if it does, what happens is the virus is able to get inside. But as soon as it gets inside and attacks the nucleus of the self cell, the nucleus changes its behavior and takes over to make new virus. But the good news is it also changes its marker system. A virus-infected cell is now going to show some additional markers on its surface. This cell now becomes a non-self cell because the sequence changed, right? It inserts new markers on the outside of the cell. This allows the immune system to recognize what was previously a self cell as now a non-self cell. So a virus-infected cell is a non-self cell. You follow me on that?
Yes.
Okay, so that new sequence on the outside of this virus-infected cell identifies it as non-self. So now it's fair game for the immune system. That's what we like about that as far as that goes. Now, there's one other cell type that we'll talk about before we get into the specific immune system. And it's got a great name. It's called a natural killer cell. NK. Now, what we know about natural killer cells is that they are really part of our innate system. And they respond quickly, really fast, which is what most of our innate cells will do. They to virus-infected cells or cancer cells. So they represent, a percentage of the white blood cells that circulate in the plasma as well as in the lymph fluids. We see them in the nose and in the spleen and places like that. And they release chemicals that are going to promote the immune response. So they have lots of overlapping responsibilities. Remember at the beginning of today, we said that the immune system was interlocking and multi-layered. So lots of these cells release chemicals that promote the actions of other cells in the immune system. But they constantly contact other cells. And really what they're looking for when they contact other cells is they're testing that other cell. Is it self or is it non-self? So in the blood or the lymph, they're going to contact other cells, and it's going to depend on a balance of signals. And sort of assesses markers on the cell it bumped into. So we can have some activating receptors versus inhibitory receptors. So when it's assessing these markers that are on the cell that it bumped into, they're either going to be activated or they're going to be inhibited based on whether it's self or non-self, essentially. So when they're inhibitory receptors, that's a self cell, a healthy self cell. And activating factors would be on cancer cells, virus-infected cells, and those will switch on. And when they switch on, they release enzymes that destroy the target. And the target was the activating receptors on the cell of the cancer cell or the virus cell. So we're destroying the target. And in this case over here, we switch off the activation because it's a healthy cell, it's a self cell. You follow me on that?
Yes.
Now, here's a little tricky thing. So these viruses, and we know this even just by paying attention to what's going on in the pandemic, these viruses are not very complete organisms. So they're very simple and they change all the time. We had a Delta variant, we had the Omicron variant, we've got the BA.1.2 variant or whatever they're calling the next one that's going to represent the 6th wave of pandemic. Right, and so they mutate and they change all the time. One way that they can sometimes change and try to evade or avoid the immune system is they can infect a cell, and rather than having a different marker on the outside of the cell, they make the markers disappear. So we can have activating receptors, or we can have no receptors. And no receptors are going to switch on the natural killer cell too. So, the viruses and the bacteria, they try to survive by doing little tricks. They try to hide from the immune system. But in the case of the natural killer cell, that's not going to work. So they infected a healthy cell, and in an attempt to avoid being identified as a virus-infected cell, they remove all markers. But by definition, that's a recognition of non-self because it doesn't have self markers on it anymore either. So the no receptors also switches on this natural killer cell. And the natural killer cell then goes and releases enzymes that are going to destroy the target. So they can release enzymes that poke holes in the membrane and allow extracellular fluid to leak in. That's going to alter the function, usually destroy it. Or they can actually release enzymes that go right into the organism, the bacteria or the virus, and degrade them, break them down. So those are the different ways that those enzymes can destroy their target. Okay, so those are all examples of non-specific immune responses. When we look at adaptive immunity, and we'll get this started today. So adaptive immunity is going to be activated early, but it requires time. So it seems like it's sitting back, not doing anything initially, but the reality is it's going through the steps necessary to specifically address that virus or that bacteria or that cancer cell. And that, because of the specificity, requires some time. It also requires some checks and balances because this is dangerous, right? These cells are all dangerous. If they started to attack normal cells, we'd be in deep trouble. And that's what autoimmune disease is. You'll talk more about that in pathology. But autoimmunity occurs when cells that would normally attack non-self cells begin to attack self cells. And we'll talk about how that's possible when we talk a bit further along in this adaptive immunity scenario. So the features of adaptive immunity that make it unique and make it really, really special are that it has specificity. So I mentioned already that the active cells here are T and B lymphocytes. So T and B lymphocytes are going to have some very significant differences. They're similar in the fact that they're specific, but they're different in the ways that they learn to be specific and the way that they act on the targets. So we'll talk about some differences. And I think in your not too distant future, if you catch my drift, that understanding the differences between T and B cells is going to be very, very helpful to you. So we'll talk about some of those differences and we'll highlight those in a minute. But what we're saying when we talk about specificity is that we have available to us T and B lymphocytes for every variation of cell marker, known and unknown, in the universe. There might be a virus that circulates on the planet Mars that has never been exposed to us here on Earth, but we've got a T cell for that, and we've got a B cell for that, based on protein markers. So we know that there's about 20 amino acids in the universe. Proteins are going to be built from amino acids. There's a group of cells, T cells and B cells, for every possible protein marker in the body, produced in the bone marrow and matured in the two locations, bone marrow and thymus. We'll talk about that in just a minute. But the beauty of this system is it doesn't matter what we're exposed to. We've got a cell that is able to recognize it. The trick, like I mentioned earlier, is the time. If it takes time, we may lose the battle. If it's a very virulent infection, can cause serious injury and illness, it may not be time to survive. right? So that's where vaccines come into play. We're trying to bypass some of that time factor there. So specificity is an important feature of this adaptive immunity. It's described as being systemic. In other words, it's not dependent on a local stimulus. So inflammation was local, right? Inflammation occurred because we damaged some cells. An infection starts as being local. But the adaptive immune system is going to give us cells that will be circulating in the blood and in the lymph and in the lymph nodes and in the spleen. They'll be all over the body. So they will be available for response in other places than just the original location of the infection or the injury. So systemic is an important factor. And the other factor that makes this system unique is memory. After a first exposure, we have memory cells in large numbers for that specific antigen. So think measles, right? Most of us have a childhood vaccination for measles. We've never been naturally exposed to measles, but we have been vaccinated for measles. The goal of a vaccination is exposure to a virus or a bacteria without getting sick. That's the goal of a vaccination, right? The goal of the coronavirus vaccination is to give us exposure so that we get T and B lymphocytes activated and create memory for subsequent exposures. Now, we've learned about the coronavirus that it's not the greatest vaccine because it doesn't prevent infection. That's what it looks like now, right? In the third, fourth, fifth waves, we're still getting infections in people that have been vaccinated. But the good news is those people are not getting as sick. So it's serving a purpose. It's not preventing infections, but it is preventing what we would call morbidity, how sick you get from the exposure. So it serves a purpose, especially in people who are otherwise compromised. When your immune system's compromised, it means you're not going to fight these infections very effectively. And as we get older, that becomes an issue. When we're very young and when we're very old, the immune system is not very effective. So we have some mechanisms available to us to make up for that. You know, mom's breast milk and the mom's blood through the placenta can give us some immunity under those circumstances. Some antibodies cross the placenta, some antibodies cross breast milk to provide the infant or the fetus with some immunity until their immune systems improve, until they mature and get effective. So these are things that we think of when we think about this adaptive immunity. Now, when we look at adaptive and specific immunity, we recognize, the first thing we want to recognize is that there's an overlap. So we've talked about some cells that we can see in this picture. So we've talked about a natural killer cell. We've talked about neutrophils. We've talked about macrophages. We've talked about some complement proteins. So all of these are represented in our little diagram here as parts of the innate or non-specific immunity. Over on adaptive immunity, we've got our B lymphocytes and our T lymphocytes, and we see that there's an overlap. There's an overlap in the way that we respond between the two systems. So it isn't a wait and see, it's throw the kitchen sink at everything. Put everything we've got available to us into preventing this infection from becoming serious. So that's what this area represents, an overlap of the two systems. This is typically going to respond within seconds. This may take days on a first exposure. But after that first exposure, it's going to be rapid. It's going to be very fast. Okay, so this gives you a visual of the sharing of responsibilities between the innate and the adaptive immune systems. Let's see here, where are we? Okay, let's go to next page. And we're going to have to add in a couple of layers here. Okay, so we're continuing on with our adaptive immunity. We recognize that it's going to be specific, it's going to be systemic, and it's going to generate some memory. So there's two pathways. One way is sometimes called cell mediated. And all you need to remember when you see cell mediated is to realize that there's going to be cell-to-cell contact. Cell, like the natural killer cell, binds to the membrane of the virus or the bacteria and releases enzymes into it. That's cell-to-cell. So that's going to be using what we call cytotoxic T lymphocytes. So once we get these two sentences in place, I'm going to stop writing lymphocytes. I'm just going to call them T cells, but recognize that they are lymphocytes. And the B lymphocyte, we're just going to call a B cell. Okay, so that's the other pathway. In the other pathway, it's antibody mediated. And that's through B lymphocytes and B lymphocytes are going to be, when they get activated, they become plasma cells, and the plasma cells are the ones that produce antibodies. So that's what we measure in the blood when we wonder whether we're protected, for example, from measles or from polio or from rabies or from the coronavirus. So we can measure antibodies because they circulate in the plasma and they're fairly easy to measure. So if we have elevated levels of antibodies for the coronavirus, we can be fairly safely assured that we've got immunity to the coronavirus in that case. But again, these are specific. So we could have antibodies for the coronavirus, but not have antibodies for measles, right? If we were vaccinated for measles and we got boosted regularly, we should have antibodies for measles. But it's specific, so we're going to have the same pathway repeating for every possible antigen. So there are trillions of T cells, trillions of B cells for specific antigens, but most of them have never been activated. Most of them have no reason because they've never been exposed. So the key thing here for these pathways to be activated is a process that's called recognition. Okay, so we need to see three things through. We need to recognize. Remember, we call that immunogenicity. An antigen has to be recognizable. And the result of getting recognition, hopefully, will be activation. And activation refers to the destruction of the antigen. Got to get rid of it somehow. And then we have to see, or we want to see for completion, proliferation. And proliferation means we're going to make new cells that are specific to the original antigen and memory cells specific to that antigen. You follow me on that? So 3 steps we want to be aware of.
Yeah, that makes sense.
Okay. So that brings us to a dendritic cell. So dendritic cells have a cell body. They have a cell body and they have these long extensions that are coming out of the cell body. I don't know why I didn't include these. So sometimes they look a little bit, they sometimes describe them, they look a little bit like a starfish. But these long extensions have receptors on them. And they're these receptors that I mentioned earlier, these pattern recognition receptors. So let's just say, let's keep it really simple. We're just going to put a sequence, and they would be all over, not just on the ends. So I'm just going to make the sequence 2. Okay, in reality, there's going to be 3 or 4 or 5 or 6, but we're just going to keep it nice and simple. Obviously, the more lengthy the combination, the more specific the antigen becomes. But this is a dendritic cell, and this represents a pattern recognition receptor. Okay, and we're going to make sure we understand there's lots of them on this dendritic cell. So these dendritic cells, they're located all over the body. They're found in mucosal tissues, and which are located in entry points into the body. So we find them in the mouth and in the nasal cavities. We find them in the throat. We find them in the sphincters associated with the anus and the urethra. They're found all over the place and in higher concentrations in places where we're going to be most likely exposed to viruses and bacteria. And they constantly sample the extracellular fluid. And they're literally looking for non-self receptors. So these pattern recognition receptors are designed to pick up anything that's not self. So these cells are phagocytes that are looking for non-self. And so they're identifying non-self. It's really a type of non-specific immunity. So they can't tell, they won't be able to differentiate between a bacteria or a virus-infected cell or a cancer cell, but they can differentiate between self and non-self. Pattern recognition, leave self alone, deal with, and by dealing with, it's phagocytosis. So when they match up with a non-self organism, phagocytosis is going to occur. Okay, they ingest the antigen and insert on its membrane new markers. So this now in effect becomes a non-self cell, but it's an immune cell, so it's going to have a different role to play. It will not be attacked by other cells, not yet. And it becomes mobile and travels to lymph nodes. Okay, so it starts off out in the mucosal tissues, out in the peripheral tissues, looking for non-self cells. When it finds a non-self cell, it ingests it and converts from being a phagocyte to being a mobile cell. And now what it's going to do is it's going to take off. It's going to travel through the lymph fluids to the lymph nodes to present to cells in the lymph nodes for presentation. And the presentation now is the connection to the specific immune system. So we started off non-specific. This is kind of the cell that joins specific and non-specific immune responses. So this dendritic cell travels to lymph nodes for presentation. That was the last line on the previous slide, so we're just sort of getting caught up. So that makes this cell known as an antigen presenting cell, an APC. So the APC shows up in the lymph node, and it's going to get jammed up, right? The bottleneck that I think it was Kara that mentioned that terminology. Is that right? Was it Kara?
Yes, it was.
So that's a great term, right? So we want to reinforce that term. It's a good visual. So the APC shows up in the lymph node, it gets jammed up, bottleneck. And that's going to provide more time for it to seek out a specific cell for this antigen. So the APC cell is looking for a specific helper T lymphocyte. So now we've put a specialty onto a type of T cell. We're going to see that we've got what are called, I used the term earlier, cytotoxic T cells. This is called a helper T cell. The helper T cells are going to be specific and located in large numbers in the lymph nodes. So there's a helper T cell for this specific antigen. So if we said this APC was specific, we'll identify it with a little yellow dot. Then this helper T cell is specific for that antigen. If we're dealing with more than one virus, we have to have more than one helper T cell. So when the antigen presenting cell matches with the helper T cell, that's recognition. And that's going to cause activation. And when a helper T cell activates, it increases in numbers. But remember, it's increasing these specific helper T cells. And then the helper T cells, okay, this is where we follow a pathway, and we're going to do this a couple of different ways. What time do we finish today? 10:40?
I think so, unless someone
Yes.
else.
Does it sound right, around 10:40? So we got about another, not quite 20 minutes. So helper T cells then are going to go in two directions. They go back to the site of exposure and promote phagocytosis. So remember those neutrophils and those macrophages, they're at the site of the original exposure. So the helper T cells go back to that site and they promote that inflammation and that phagocytosis that was already started. And they also stimulate, or let's, a better word than stimulate, is they're going to recruit B and T cell activation and proliferation. But remember, these B and T cells will be specific to our antigen that we picked up in this at the site of the infection. So we went from the site of the infection, traveled through the lymphatic vessels to lymph nodes. Some cells go back to the site of infection to provide a signal to reinforce what's going on, and other helper T cells recruit B and T cells for activation and proliferation. So I've got us a diagram, I think, way back. Let's see where it is. That kind of shows a bit of a visual of that. Let's see if it's not that one, not yet, not that one. Not that one, maybe this one. No. This one. Okay, so let's get the writing out of the way. So when we're looking at this picture, we can see some of an illustration of some of our innate responses. So we've got a barrier here that's going to provide, in this case, it's an airway. This is actually an article that was talking about the coronavirus specifically. So this represents cells in the airway, in the lungs. Okay, so we got a barrier of cells that's usually pretty good. But in this case, we got an infection. And when we got an infection, the virus replicates and copies of the virus are made. So there's our nice little spike protein virus. The infected cells release chemicals, that's a fancy word for chemicals, cytokines, that signal alarms for other cells. That would be interferon. Now, they also signal for reinforcements like macrophages and dendritic cells at the site of the infection. Macrophages are going to engulf the virus through pattern recognition. Okay, and when we look over here at the same target, the same inflammation, here we have the coronavirus, the inflamed tissue, inflammation, phagocytosis, neutrophils and macrophages will be involved. The dendritic cell picks up the virus, carries it to a lymph node. And when it carries it to a lymph node, we've got the immune cells located here, B cells and T cells. B cells, T cells. When we activate the T cell with the help of the helper T cells, okay, that is a step that they've skipped in here. Well, here they show you the helper T cell right here. So this is all occurring in the lymph node. This would be in the node. We activate cytotoxic T cells. And when cytotoxic T cells are activated, they are going to target the virus-infected cell directly, release enzymes to kill it, okay, and destroy the virus that way. And the additional pathway, the other pathway, is to activate B cells. When we activate B cells, we produce plasma cells that make antibodies. These antibodies are specific to the original virus, the original antigen. And these antibodies can bind. We'll talk about what antibodies do probably now at the beginning of next week. So we get recognition, activation is what we're seeing in these steps here, and we create memory T cells, memory cytotoxic T cells, and memory B cells and antibodies. All of these things will make us better at the next time we're exposed to the virus. So we can draw it this way. We can get ourselves a nice little schematic going by watching what happens this way. So we're going to have our antigen. So our antigen, we're going to try and stay consistent and go with yellow. The antigen is going to be on a first exposure. Okay, so first time first exposure to our antigen. Okay, so our antigen is going to be identified by our dendritic cell. Now there are other ways to present, but this is the most common one. So the cell is out looking for these antigens anyway. So now our dendritic cell has displayed the antigen on its membrane, and it's going to travel. So it now becomes our antigen presenting cell. And it's going to travel to a lymph node where it's going to, and again, we'll try and stay consistent here. It's going to match up with a helper T cell and form a bond. Okay, so we're going to form a bond with our APC, a complex. And when we create this specific helper T cell with this specific APC, antigen presenting cell, we activate those helper T cells. And when we activate the helper T cells, we're going to watch the helper T cells oh, one too many erasers. Okay, now, so one thing that's going to happen is that the helper T cells are going to stimulate B cells. When they stimulate B cells, and these are B cells specific to our original antigen, when they stimulate the B cells will be activated into plasma cells, and the plasma cells produce antibodies that are specific to our antigen. And then the B cells.
So just a quick question, sorry. So B cells become plasma cells, which then produce antibodies, correct?
That's right. So the B cell circulates as a B cell, and then unless it's activated, that's all it's going to be. It's just a cell in circulation. And when it's specifically activated, it becomes a factory for producing antibodies. And additionally, it's going to make memory B cells. The memory B cells are the key really to longevity. Antibodies will often circulate for decades, but need to be reactivated on a subsequent exposure. So remember, we're talking about a first exposure. So all of this may take several days in order for us to get to the point where we actually produce antibodies that can do something specifically with these specifically with the original antigen. So we're also going to make memory helper T cells, so that these things, the helper T cell is technically not necessary. Like the B cell can be activated by free antigen. So we can do this too. We can see that the B cell could be activated directly. So in other words, if the antigen comes in direct contact with a B cell, we can get this activation. But with the helper cell step, we get a boost. We get much more vigorous responses. So we can have a direct pathway. Put it in brackets, put it in a gray color to indicate that it's not the primary, it's not the ideal exposure. We want the reinforcements from the helper T cell. Now, the helper T cell also will stimulate the cytotoxic T cells. So let's see, what color can we make? We'll make them turquoise. Cytotoxic T cells. Remember, the T cell and the B cell, they're all lymphocytes, right? So the cytotoxic T cell is going to be activated. And when it gets activated, it's going to inject this is sometimes described as cell-to-cell contact, right? When we activate the cytotoxic T cells, they're going to go directly after the antigen. And we're going to get memory T cells. Remember, these are the cytotoxic T cells. So that's a key to this specific type of immunity. Now, we can also activate cytotoxic T cells when the antigen is displayed on a virus-infected cell, for example, or we could even throw in a cancer cell for that matter. So we can have a direct stimulation too, but the ideal pathway involves these helper T cells. So that's our central pathway. Helper T cells stimulate the actions of both the cytotoxic T cells and the B cells. Okay, so this is sort of the pathway that we would see in response. So the T cell pathway gives us a specific defense against intracellular pathogens like virus-infected cells and cancer cells. So they're found within cells directly. And the B cell pathway defends against extracellular pathogens by binding to these antigen-presenting cells. They were found originally in the extracellular fluid, picked up by the dendritic cells, and delivered to lymph nodes for stimulation. So the antibodies antibodies are sort of Y-shaped structures. They're proteins, and they've got a Y shape to them, and on extensions, they have receptor sites. So in our example, they would have receptor sites for this antigen. So what they're able to do, antibodies operate by different mechanisms, and that's where we'll pick up next week. So can somebody who's going to be here next week do me a favor? This part doesn't need